Good afternoon, everyone. Thank you for joining us. My name is Marissa Waterman, and I'm the Marketing Director here at the Academy. Today's webinar, titled When Persistence Doesn't Pay Off, Integrative Approaches to Understand and Predict the Hazards of Poorly Studied PFAS. This is the second of 4 webinars in our PFAS series. Our Executive Director, Dr. Daniel Other, will serve as the moderator and will introduce our speaker today. During the presentation, you'll be able to submit a question by clicking on the Q&A icon on the bottom of your screen. We'll do our best to address each question at the end of the presentation. Before we get started, Dr. Other would like to say a few words. Dan, how are you today?
I'm fantastic. Thanks, Marissa.
Thank you.
Happy New Year to everyone. This is our first webinar of 2025, and this is the second webinar in our series of 4 webinars focused on PFAS. The Academy is excited to be able to offer a series of webinars on a focused topic. As many of us are aware, PFAS is an important group of chemicals. Our profession is working to better understand the risk of PFAS and to protect the public and the environment from those risks. And to support you in your efforts, we've put together a series of webinars which speak across various media we experience as engineers, like soils and water and air, as well as using different scientific approaches, like toxicology. And that's part of the value proposition of our Academy. We strive to be the home for environmental engineering and environmental science professionals. Our goal is to support you in your work. And of course, one of the ways we do that is by technical programming, such as today's webinar. And I also want to be sure to remind you of another way we support you in your work, and that's through board certification. Board-certified environmental engineers and environmental scientists are among the best of the best in their work. So if you're a member of our profession, and especially if you're a member of our Academy, we encourage you to seek out board certification and let us know what we can do to help you. Today's exciting webinar is brought to us through the generosity of our patrons. Patrons are organizations who see tremendous value in the work of the Academy and provide financial support. $6,000 from utilities and $8,000 from private firms every year. This helps us put together exceptional programming for our members. So please join me in thanking our patrons. And if you have an interest in knowing more about how your organization can become a patron, please let us know. And now, with all that background out of the way, let's get started. Today, I'm thrilled to join you to moderate what I know will be an exceptionally informative webinar entitled When Persistence Doesn't Pay Off, Integrative Approaches to Understand and Predict the Hazards of Poorly Studied PFAS. Our speaker is Dr. Carla Ng, an associate professor in the Department of Civil and Environmental Engineering and the Fulton C. Noss Faculty Fellow at the University of Pittsburgh. Carla enjoys secondary appointments in environmental and occupational health, as well as in chemical and petroleum engineering. She received her doctorate in chemical and biological engineering from Northwestern University in 2008. Carla's research focuses on the development of computational and in vitro approaches to evaluate the fate and the effect of legacy and emerging chemicals in organisms as well as ecosystems. And she has a particular focus on PFAS. Through interdisciplinary, cross-sector collaborations, her group works to reduce the use of hazardous chemicals in products and processes to protect public and ecosystem health. It's truly my pleasure to introduce Dr. Ng for this webinar. Before we begin, and as a reminder, please put your questions in the Q&A function, and I'll use these as part of a moderated discussion at the end. And now, with those instructions out of the way, Carla, my friend, the stage is yours. We are so excited to hear from you.
Thank you so much for that nice introduction, Dan, and hello to everybody out there in the Zoomosphere. I am excited to be sharing some kind of hot off the press work with you all today. And so let me get started with talking about the persistence problem with PFAS, which, as we know, is one of their biggest problems. And many of us working not just with PFAS, but just in general with persistent pollutants and emerging contaminants sometimes get the impression that it's a Sisyphean task to try to tackle these chemicals. And so I'm hoping what I can bring are some ideas of how we can integrate different approaches to try to get a handle on this global issue of PFAS, and particularly with respect to understanding their hazards and potential toxicity. So before I dive into that, I'm just going to give you a little bit of an overview of my group for those of you who are less familiar with it. We do work not just on PFAS, but right now we've been working mainly on PFAS. But in terms of the broad categories that my group works on is we do a lot of toxicokinetic modeling, which is basically viewing an organism as a reactor and using those series of linked reactors to predict where chemicals go in the body, what tissues they distribute to. And this can be really helpful in guiding, for example, if you're doing monitoring study, which species, which tissues you should be sampling in order to protect an ecosystem. We do a lot of molecular modeling. I'll be talking a bit about that today as a means to understand and predict the toxicity and also perhaps design ways to remediate and degrade forever chemicals. I'm not going to talk much about remediation degradation today, but feel free to follow up with me if you're interested in that. We do a lot of work in my group on human exposure via food. So these are the lectures that I put together to scare people that there's nothing out there that's safe to eat. And we've looked at a variety of chemicals in that context, brominated flame retardant, pesticides, and also PFAS. And in the region here in Western Pennsylvania, we do some work on contaminated communities and understanding different types of exposure. So for example, we've done work with a community in McKeesport that had AFFF contamination introduced into the drinking water system through a fire hydrant. We've also done a little bit of work in East Palestine to try to understand the implications of the disaster, the derailment disaster that happened there a couple of years ago. We also do faraway work. We have some collaborations going on in Ghana and Suriname to try to understand the PFAS footprint there, as well as some collaborations with folks in Samoa and American Samoa. Today, I'm really going to focus in on this, trying to understand the distribution of PFAS and their potential effects within organisms, and how we can use both in vitro and in silico approaches to get into that. So when we start to think about PFAS as a global issue, the thing that, of course, people are very concerned about is the potential toxic effects. And with PFAS, this is a big concern, not only because they are kind of ubiquitously present inside of everyone's bodies and also highly persistent, but also because they have a wide variety of recognized and identified toxic effects that cover a multitude of different organism systems. So they've been linked to things like thyroid disease, increased cholesterol levels, circulating cholesterol in the blood, liver damage, kidney cancer, testicular cancer, delayed mammary gland development, reduced response to vaccines. This is one of the most sensitive endpoints that have caused these very, very low health advisory levels that have been published, lower birth weight, and then some observations that have lower certainty. For example, some incidences of breast cancer, potential links to inflammatory bowel disease, et cetera. And there's a lot already there. They are known to disrupt a lot of different systems. And in addition to that, the IARC recently classified PFOA as carcinogenic to humans and as potentially carcinogenic, or PFOS as possibly carcinogenic to humans. This has deep implications for what we're going to do about remediation, for example, because there's still a lot of PFOA and PFOS out in the environment, even though they've been largely phased out of many uses in the US. And there are these other effects for which we're still collecting data. The thing to recognize here is that the compilation of this information has taken decades. Many animal studies, many epidemiological studies, and yet, as I'm going to show in a couple of slides, a lot of these observations are limited to a pretty narrow number of PFAS substances when compared to the total structural diversity that there is out there. And some of the things that kind of allowed us to start to derive theories about why it is that PFAS have so many of these different impacts on organisms has to do with where they end up in the body. And so a very classical paper that I often bring up in my talks, even though I tell my students not to make pie charts, I find this one very useful. This is from a Lutz Arndt's paper in 2009 that's looking at the total PFAS, that this was back in the day, so they were still calling them PFCs, body burden in harbor seals. What's interesting about this is that it's looking at the distribution in the body, not as concentration, but as total mass found there. So the percent mass found in each of these compartments. And what you can see is that, as we know for the well-studied PFAS, the majority of them are found in the blood and in the liver. And so you can see 38% here in the blood, 36% in the liver. And when you compare it to something like the muscle, where it's only 13%, now when you think about the relative size of those compartments, the muscle is by far a much larger component of the body and yet has a much smaller percentage of the total PFAS. So In addition to that, there is the understanding that high fat compartments like blubber has a very small percentage, and this is very different from your traditional POP. So this has already been known for a long time. People, understand that there's something else going on with PFAS that cause them to have this special distribution, and also that this special distribution could be potentially the source of their impact on multiple metabolic systems. If you look at a much more recent study, this is from Anna Roebuck in 2021, looking at birds from the Cape Fear River estuary. And of course, the Cape Fear River is one of these highly contaminated areas due to the Commerce plant that was emitting legacy and novel PFAS over its history there. What's interesting here is that if you look at the tissues of these birds and you look across some quite well-studied PFAS like PFOS and some more emerging PFAS like this Naphthalene byproduct 2, which is on this replacement ether type of chemicals, you see that those distributions can be quite different. For example, you see for Naphthalene byproduct 2 that actually you do get some accumulation in the fat of the birds, where you don't see that very much with the PFOS. You also see other things pop up, like for the very long chain PFAS, we find greater accumulation in the brain, and that's been replicated in other animals, including pilot whales and polar bears, as well as some observation in human patients, human cadavers, that long chain PFAS can accumulate in the brain. So we find that different PFAS have specific preferences. So the patterns of PFAS distribution in different species shows specificity, a preference for specific tissue and components, and selectivity, so different patterns for different PFAS. And so even if we have some very well-studied PFAS that can tell us about mechanisms of toxicity, there are obvious gaps for PFAS that are less well-studied that may have different effects, due partly at least to their different distributions and their opportunities to interact with different types of receptors in the body. Oh, looks like I messed up my animations here. So what can these molecular interactions, down at the tissue scale tell us? So where many of us started who are working on PFAS bioaccumulation and understanding PFAS bioavailability is to look at the analogies between the perfluorinated alkyl acids, which are the best studied classes of PFAS, and the non-fluorinated alkyl acids, which are fatty acids that are really important in our normal body function. So if you look at a PFOA molecule and you look at an octanoic acid molecule, they're the same thing, except you've replaced all the hydrogens here with fluorines in this alkyl tail. And so many of the same receptors that interact with fatty acids in the body also interact with PFAS. We know that for fatty acid carriers in the blood, like serum albumin. We know that for fatty acid binding proteins within the cells, like liver fatty acid binding protein, they also bind to PFAS. We also know that there are membrane-bound transporters that are meant to transport short-chain fatty acids, and they also transport PFAS. And these are important because they help to mediate elimination rates, so uptake and efflux into and out of different tissues. That applies to the kidneys, that applies to the gut, and concerningly, that also applies to the brain. And then in addition to this, we know that the structure of fatty acids and the structure of PFAS, at least these perfluorinated acids, are similar to the structures of the things that make up our cell membranes, which is a really integral part of all of our bodies. And so things that can intercalate into and potentially disrupt these membranes can have really negative consequences. So understanding the interactions of PFAS with membranes is really important to understanding not just their distribution and accumulation in the body, but potentially some of their important toxic effects. But still, we don't have data for many PFAS, right? So those perfluorinated acids are sort of the model compound that we kind of base our PFAS intuition on, and yet there's a lot we still don't know. So this is a figure that I found really kind of astounding because it was published in Clean Water in 2023. So a very recent publication that shows that most toxicity studies still have only focused on two PFAS, PFOA and PFOS, the two that are no longer in active commerce in the US and much of the rest of the world. They're still making up almost 90% of the toxicity study. Fewer than 15% address the thousands of other PFAS. And in addition, there's been a preponderance of studies on particular types of toxicity, but much less on other types of interactions of PFAS with body systems. So as of this 2023 review, cell viability, endocrine disruption, reproductive, and metabolic effects were the most studied endpoints. And another kind of interesting data point here is from another recent paper in Science Advances that looked at a retrospective analysis of PFAS that were in human and wildlife samples. So they basically went and looked at bank samples and analyzed it for legacy, emerging, and unreported, so newly kind of discovered PFAS. And this kind of shows that the unreported PFAS, this green part, is a large part of what's actually found in not just different biological samples, but also in different environmental samples. So this means that not only are these PFAS, constantly changing what we're being exposed to as PFAS are phased out and new PFAS are brought in, but some of these unreported ones may have been around also for decades and we just didn't really know about them. This is kind of what has emerged by looking into these biological samples that are a really important repository of kind of our unknown unknowns. So in my group, we're really interested in trying to bring together as many different types of lines of evidence as we can to kind of try to understand the relationships between PFAS in different tissues, across different organisms, uptake, comparisons, who's a good model organism, can you use a fish to look at human toxicity, is a mouse or a rat better? So we're trying to answer questions like, why do PFAS bioaccumulate? Why do they have preferential tissue distribution if they do? And how does this impact their toxicity? So I'm going to talk about two kind of hot off the press studies today that were led by one current grad student and one student who just graduated and is starting a short-term postdoc in my group. So the first one is looking at an in vitro study done by Ruiwen Chen, who's a current graduate student in my group. And we started from this idea that, prior to the whole PFAS issue, KOW was like the tried and true metric by which people used to kind of do the engineering estimation of, is this compound bioaccumulative? How much of a problem is it going to be? Can we estimate narcotic toxicity on the basis of this? And that KOW metric really failed for PFAS, especially when you consider the pKa of the acids. And so we started to think about, okay, KOW doesn't work on its own, but as a concept, this partition coefficient is really useful. And we all have kind of the infrastructure to be able to deal with understanding partition coefficient. So maybe it's not so much just throw out the idea of a partition coefficient, but how do we make one that is appropriate for understanding the behavior of PFAS in organisms? And so we took this PBPK framework that we've developed in our group to try to understand all the interactions that happen within each tissue for PFAS, which our previous models had done in a way that's quite complicated, many, many parameters, because we're looking at the specific protein binding in each type of tissue, and we look at transporter interactions, leads to many parameters. And we wanted to simplify that into a sort of partition coefficient structure. And so our goal is to move from a two-phase partitioning model to a multi-phase partitioning model where we come up with a tissue fluid partition coefficient. Let me pull up my little laser here so I can point better. A tissue fluid partition coefficient that includes different classes of phases within a tissue. And those phases we've broken down into 4 major categories. Structural proteins, thinking about like the types of proteins that make up your muscle. Functional proteins like serum albumin or other carrier proteins that are meant to be shuttling things around the body. Storage lipid, which is what you find in your adipose tissue, and phospholipid, which is the components that make up all of our cell membranes, and it has quite different character from neutral storage lipids. And we put all of those together in kind of a weighted partition coefficient that is the partition coefficient to the particular phase times the fraction of that phase that the tissue is made up of. So we're kind of starting on this journey to make this new multi-phase partition coefficient. And what I'm going to focus on for today is just 2 components of that. the phospholipid and at least one of the functional proteins for the first half of the talk, and then I'll move on to talking about other receptors and their links to toxicity. So this paper was just accepted over the break, so it's just out now in these past couple of weeks, and it's all about deriving both membrane water and protein water, I'm going to call them partition coefficients, from in vitro experiments. And this was a collaboration with Derek Minsterman and Jennifer Field at Oregon State. And we were able to make laboratory measurements of phospholipid membrane partitioning and human serum albumin binding for 60 structurally diverse PFAS. So we're basically taking the membrane water partitioning approach that was founded by Stephen Droge. He had some foundational studies that were published on this, and expanding that data set out to more structurally diverse PFAS, and then also adding and comparing to that the albumin binding data. So basically we're filling in the purple and the red part of this tissue fluid partition coefficient. For those of you that are not familiar with these assays, for the membrane water partition coefficient, so this is the phospholipid, PFAS phospholipid interactions, we use what's called a solid supported lipid membrane assay, which basically consists of these little beads on which they have put basically a membrane phospholipid. And then you do a series dilutions of these beads. So you have different concentrations of phospholipid membrane in each of these tubes. And then you add your PFAS and you let that equilibrate. And then you measure how much PFAS is left free in solution after equilibrium. Similarly, for the serum albumin dialysis experiments, we use these handy little dialysis cups that have a membrane at the bottom. And so you can fill this system up with your PFAS solution, and inside of the cup you have your membrane, and it cannot diffuse through the molecular weight cutoff membrane that's here. And so the PFAS equilibrates freely across this, and you get your equilibrium affinity constant or your equilibrium dissociation constant, depending on how you do the math. So just to show you kind of what goes into these calculations before I show you all the results, the solid supported lipid membrane, so the phospholipid water partition coefficient, is fairly straightforward. There you're just kind of exposing your PFAS spiked water to the membranes, and then you're going to measure at equilibrium the amount of PFAS that's on the membrane versus the amount of PFAS that's in the water. And the way that we do that is by subtraction. So we can't actually measure what's on the membrane. We just measure what was there before and what's there at equilibrium. And by difference, we decide what is bound to the membrane. And this has to be said that this works great until you get to the really hydrophobic PFAS. And then the really hydrophobic PFAS likes to stick to everything. That includes the surfaces of your container, and it includes the beads on which the phospholipids are secured, and sometimes not very well secured. So you have to account for this by doing extra rinses in your assay system. And then for the serum albumin binding, the binding is done using a binding isotherm, where the affinity constant is just the ratio of the on and off binding coefficients. And you do this by making some assumptions about the number of binding sites that are on the protein. And then from that, you get a binding affinity or a dissociation constant. They're just the inverse of one another. And I think this is one of the more important things that I'll come back to later. This is a binding constant, right? So it tells you what the specific binding affinity of the PFAS is for your protein. It is not a partition coefficient. And it is saturable, which means that this value will change according to how much PFAS is in the system relative to how much protein is in the system, unlike the membrane water partition coefficient. So we have to somehow convert this affinity constant to a partition coefficient, which we're calling a distribution coefficient between human serum albumin and water. And so I will talk about what the implications of this are as we look at the results. So let's look at some results on what we found for the membrane affinity of different PFAS and different PFAS classes. So first, I will point to the studies by Stephen Droge that did some of the first measurements of this for PFAS, which we kind of jumped off of to do this study. And you'll see them noted in the clear symbols on this graph. And then all the colored ones are the ones that we filled in. So you can see that we went to quite a bit longer chain length. We went up to the C16 carboxylic acid. So on this plot, we only have membrane water partition coefficients for per and polyfluoral carboxylic acids, and I'll show you the other groups after this. So on the y-axis, you have the log of the KMW, and on the y-axis, or the x-axis, you have the fluorinated carbon atom number, so the number of fluorinated carbons present in the molecule. So just to point out a couple of different things, in general, our results agree with Droge's results. We're just kind of adding more groups and extending out the chain length. And we found an increase of 0.36, that should say log KMW per fluorocarbon group. So this means that for each increase in the fluorinated carbon chain length, you get an extra 0.36 log units in the phospholipid water partition coefficient. In general, the PFCAs and the FTCAs, so the blues and the reds here, increase with chain length. Although you'll notice that you have these parallel lines, but with different intercepts. So the N2 FTCAs, the fluorotelomere carboxylic acids, and the fluorotelomere unsaturated carboxylic acids are on a different intercept, which just means that while the increase with the fluorinated carbon number is the same, they have a lower membrane water partition coefficients than the perfluorinated alkyl acids. And we didn't see any such relationship for the ether carboxylic acids. You see it's essentially a flat line here. So they behaved quite differently from the other compounds. For the sulfonate compounds, we have a greater diversity of groups present, and you'll see that there's kind of a variation in the intercepts here, although the slopes, again, all look quite similar. And we see a very similar slope than we saw for the carboxylic acids. So per fluorocarbon unit that's added, you again get a 0.37 rather than a 0.36 log unit increase. So the increases are very similar. However, the intercepts again are different. So it's the intercept that tells you which functional groups have the strongest affinity for the membrane. And the sulfonamides and the fluorotelomeres are falling at the lower end of the intercept, suggesting that perhaps because of their lower surface activity, that also lowers their membrane interactions. Among the C8 sulfonates, which is interesting, so if you're just looking at the C8s here that are all stacked up, PFOS clearly has the highest KMW, whereas the cyclic compound that I've shown here, in case you're not familiar with it, this is an ethyl cyclohexane has the lowest membrane water partition coefficient. And then we had a few other ones that are sort of in the other group because we didn't have enough molecules to really put together a clear relationship. So we can't say much about how they behave other than clearly the high ones here are the dipaps. And if you're familiar with PFAS, it makes sense because dipaps have these two fluorinated tails, and so they have a very large number of fluorinated carbons in their structures. And so this is just showing the A2 dipap as an example of this. Now, when we turn to the protein binding, the picture actually looks quite a bit different. So as we saw with, let me just go back and kind of remind you, for the carboxylic acids, it's quite clear that as the chain length goes up, the membrane water partition coefficient goes up. So it's very much driven by the number of fluorinated carbons in the molecule. Similar can be said if you look at the PFSAs, it's just clearly increasing with fluorinated chain length. But now when we look at the binding, you can see that for the HSA, the clearly highest binding affinity of all of them is PFOS. So if we compare that to the PFCAs, we know that the PFCAs go to very long chain lengths, but again, the strongest binding for the PFCAs is PFOA, is the C7. So there's something that about PFOA and PFAS that says they're kind of at the sweet spot for maximized binding with HSA, which kind of makes sense in terms of what we know about their toxicity. Whereas there are some quite longer PFAS that have relatively lower binding affinity. So PFAS has the highest affinity for HSA across all groups and all chain lengths, including the longer chain sulfonates for these groups that are well represented. Now, I don't have all the groups here because some of them didn't have enough to make a box plot of. And so on the right, there's a little bit of a focus just on the C8 baseline, so things that are on the C8 backbone. And you can see again that PFOS is one of the strongest, but actually just beating it out, which was not on the other plot, is the 8 chlorinated PFOS. So if you add a chlorine to that PFAS backbone, then you increase the hydrophobicity and you get even a stronger binding to HSA than you do for PFAS by itself. And you see the same thing happen here for the chlorofluoroether. That's the 9-chlorine PF3ONS, which is a mouthful. So I put the structure down here so you can look at it. It's basically just a PFOS where you've added an ether linkage there after the 2 in from the head group, and you've also added a chlorine. And that makes its binding affinity get closer up towards the PFAS. Okay. So in addition to looking at that, we wanted to see, well, what can docking simulations tell us about what we're seeing in vitro? Do we get any additional insight from this? And also because a lot of work in my group is doing these molecular simulations, and we're wondering how good are they in terms of, can we compare them to experimental work? And that'll come up again in the next section. So what we found is that docking, which is a pretty rapid means of molecular simulation, works pretty well up to an association constant of a log Ka of 6. And after that, the docking simulations continue to say it increases, keeps going up, but the in vitro work says, nope, that's where it turns down and starts to go down. So remember what I said, that PFOS was kind of the sweet spot for maximum serum albumin binding, and after that it decreases. The docking analysis is missing that decline, and it just says it should keep going up. We also try to do some correlation of the binding site for where these PFAS are binding on HSA, and comparing that to the known drug binding sites, so and fatty acid sites that are present in HSA. And other than PFOA and PFOS both going to the same fatty acid site 6, we didn't really see any kind of relationship that clustered together types of PFAS with particular binding sites. So we didn't really see any clear relationship here for the docking analysis. So when we then directly compare our membrane water partition coefficients and our human serum albumin affinity measurements, we kind of get this, what I call like a gunshot scatter plot, which means there is no relationship between the x-axis and the y-axis. You know, we've tried to color code them for the different types of PFAS, and there is not a relationship. But this is actually good news, because it means that what's happening on the membrane and what's happening on the proteins are mechanistically different. This is not redundant data. So these are complementary data that are telling us that both of these things influence where PFAS will end up in the body, but it is not that you could get one from the other. So you need both in order to understand what's going on. And this is a really hard plot to interpret, so I'm going to walk you through it because I think it's one of the coolest things to emerge out of this comparative study. So on the y-axis, we have the HSA water distribution coefficient. So now we're no longer talking about the affinity for binding, but we've translated this into a pseudo partition coefficient. And on the x-axis, we have the phospholipid membrane water partition coefficient, which is a true partition coefficient. And what I want you to note is that this solid line here is the solid line at which you would have equal preference. If the PFAS was in water, it would be equally distributed to either HSA or membrane. And as you move to the right, then it is preferring the membrane. And as you move to the left, it's preferring the protein. More interesting is if you look at these colored bars, the colored bars are prediction of where the PFAS distribution would be in this 2D plot, depending on what the exposure concentration of PFAS in the water was. And I'm going to specifically point you to PFOA, PFHXS, and PFOS, where you can see these bars. So the bars are actual minimum, maximum, and mean serum concentrations for human populations where it's actually been measured. And so what you can see is that PFOA concentrations in human serum are measured anywhere from the low nanogram per milliliter concentration. This is the background US population number, up to 10,000. And this is for highly exposed occupational exposure. For PFHXS, it's mostly up in this low nanogram per milliliter, and for PFOS as well. So our available data for PFAS in the public have a very wide range for PFOA because of occupational exposure, but for other PFAS are generally in this low nanogram per milliliter concentration in blood. And at those concentrations, the preference for the PFAS is to be more distributed to HSA. And as you move down into higher concentrations, it shifts from being more distributed to HSA to being more distributed to membranes, because the HSA essentially becomes saturated. And then the leftover PFAS would move more into the membrane distribution. So this potentially has important implications, one for occupational exposure, because it's saying that if you're exposed at very high levels, you may have a fundamentally different internal distribution because of this saturation of protein binding. And two, that we need to think about how we're conducting toxicity experiments if we're conducting them at very high concentrations, whereas low concentrations may lead to a different internal distribution. So what we found from these in vitro assays was a consistent relationship between an increase in the membrane water partition coefficient and the number of fluorinated carbons in the molecule. So that's about a 0.36 log unit increase per additional FC, and this is given to you by the slope of those curves. The intercepts of those curves, which varies across the PFAS groups, make the difference across these functional groups and types. So for example, it explains why sulfonates have a higher membrane water partition coefficient than a carboxylate does for the same number of fluorinated carbons. But HSA is different. So both in terms of the chain length relationship and saturability, for chain length and functional group, there appears to be a sweet spot. So for example, PFOS shows maximum affinity. And you really need to consider the interplay of both of these in order to understand the influence of exposure dose. But what does this mean in terms of the so what question, right? So the exposure question is really important question, but what a lot of us are worried about is what does that mean? And so I've taken more time than I was meant to in this first part, but I'm going to walk you through another paper from a recent PhD graduate in my group, Yushin Kao, that tries to understand what molecular interactions might mean for toxicity, and how we can generate data when there are no standards and no way to run experiments on many of these PFAS. And so what she's trying to understand is how to use molecular interactions to categorize hazard by identifying specific molecular interactions, for example, like molecular initiating events within adverse outcome pathways that can inform toxic mechanisms for chemicals. So looking into, for example, the major reported types of toxicity for PFAS, you can identify a number of different receptors that are associated with those effects. But the problem is we don't have data for most PFAS, and we don't have data on specific assays for many of the proteins, right? So for example, the estrogen receptor, ER alpha, there's a lot of in vitro data available for that, but other receptors like TTR, very little out there. So how can we start to generate some of that data? So her approach is to generate the data in silico and then use that as a means to rank the potential hazards of different PFAS. So this paper was accepted this week. So I don't have a DOI for it yet, but stay tuned. I hope to be able to share it soon. And it's a specific case study to look at PFAS that are used in photolithography and try to understand which of these PFAS might be hazardous that have never been tested before. And using a specific approach to try to reduce the computational cost of doing these simulations. So we identified 221 different photolithography relevant PFAS that are used in many different types of applications. Please go and look at our SI for the full list. And we selected 96 of these to model. And we modeled these 96 different PFAS with 5 different biological receptors that are relevant to bioaccumulation and toxicity, and across 4 different species to compare model organisms to humans. So we looked at liver fatty acid binding proteins, serum albumin, PPAR alpha, PPAR gamma, and transferrin. And we did this using this approach called the relaxed complex scheme. The relaxed complex scheme is a means to try to do molecular dynamics on the cheap. So normally we want to do molecular dynamics because it allows both the protein and the ligand to take on different conformations and then find the real minima in the energy. But that takes a lot of computational effort. And so the relaxed complex scheme does this for just the protein, which means you only have to do it once. And then you select snapshots of those protein conformations and you dock them against your many ligands. So in this case, our PFAS. So that really speeds up the process quite a bit. And then we compared that method to the docking method alone to see whether it really helped, and then ranked the interactions that we found. So I'm just going to kind of give you a little bit of the highlights of this, and then I'm happy to talk more about it. So this is how we did validation. We basically identified the receptors for which we had more in vitro data, and then compared our relaxed complex scheme with docking alone. And I'm going to zoom into one example of this so you can see what the difference is. So the squares down here are molecular docking by itself. And you can see, remember what we saw in the docking that I tried to do in the previous study, where it just kept getting stronger with chain length, and that didn't actually follow what you saw with the in vitro results. And you can see that for human liver fatty acid binding protein and also for human serum albumin, which is what we were working on in the last paper, with the relaxed complex scheme, you now start to recapitulate this falling off of the affinity as you get beyond about a C8, and then it starts leveling off. And so we think that this method is really much better capturing what you see in real life. And so you see both the relative, so the shape of the curve is closer to what you see in vitro, but also the distance between the in vitro and the modeled curve. So the blue is this relaxed complex scheme, gets smaller relative to the gray. So we are improving our ability to make predictions while not making it so computationally expensive. And then if we look at the different receptors and the many different groups of PFAS that we looked at, these all kind of look similar in the sense that there are these sort of different shapes for the different groups. I don't want you to stare into this and get too much out of this, except to say that a lot of it has to do with the chain lengths that are represented. The longer the chain length, to some extent, the higher the affinity, and some of the represented groups were shorter chain length than other represented groups. The one thing that I will point out, and that I'll highlight in the next slide, is across all of the receptors except for the liver fatty acid binding protein, you see this group that is down below that horizontal line. The horizontal line represents the affinity for PFOS, which we know is a tightly binding PFAS. And there's one group that is consistently even tighter than PFOS, and those are the side chain fluorinated aromatics, which don't sound great or not great. So we did an analysis across the different receptors to see which of these PFAS bind more tightly than PFOS. And all the deeper blue you see, the more tightly bound it is than PFOS. The ones that are blank means they didn't bind as tightly as PFOS, so we ignored them. And here is just like a sample of the ones that bound most tightly. And you can see there are many of these side chain fluorinated aromatics that show up there. So we found high affinity PFAS dominated by those that have very long chains or many CF groups, aromatic PFAS, and there was one cyclic PFAS. And the higher affinity of these PFAS was observed across all of the receptors except for LFADP, which we think is because that receptor has a small binding pocket and so is not able to fit all of the same PFAS. But this is about all we can say at the moment because these are largely untested PFAS. They don't have standards available to carry out experimental assays, but based on these results, I think they really warrant some prioritization. We also looked within the groups to understand whether branched or cyclic structures changed the PFAS. So we looked at groups that had both linear and branched, and it really varies across the groups. Sometimes the linear ones are stronger binding, sometimes the branched ones are tighter binding, sometimes the linear are tighter binding, sometimes the cyclic are tighter binding. So it seems to be specific to the groups and specific to also the chain lengths within the groups. Not much we can say really about that. And the other thing we looked at was whether the binding position could help us figure out hazard. So in the top row here, we have our different receptors and their specific binding sites are kind of shown in these red bubbles. And then in the middle row, we showed the binding sites of the most strongly binding PFAS. And in the bottom row, we showed the binding positions of the most weakly binding PFAS. And again, we didn't see a lot of different patterns here. We basically saw that all of them distribute through the binding sites. But interestingly, the most strongly binding PFAS actually had quite a few that were outside of the main binding sites. And to us, that said perhaps that the more strongly binding PFAS also have non-specific interactions as they get more and more hydrophobic. So from this very fast look at the second study, we basically showed that predicted protein binding affinities were influenced by structural features in PFAS, including the fluoridated chain length, the molecular size, and the presence of aromatic rings. They were also affected by the dimensions of the protein binding pocket. So we saw some limitations in the smallest protein that we modeled, the LFADP. And we notably identified 22 PFAS that were predicted to bind more strongly than PFOS, suggesting that they have potential for bioaccumulation and adverse biological effects. But this needs to be further validated with outcomes of protein binding. This is only one component of a complex cascade. Adverse outcome pathways can be quite complicated and long, and so we need to have more information to see whether this binding event is going to lead to an outcome at the organism level. So in terms of overall take-home messages, because I want to leave time for us to have a good conversation, there are still a large number of untested PFAS across many categories of use, and because of this use, we know that there is human exposure. And the modeling strategies, like I showed here, allow us to increase throughput on PFAS evaluation, but they're not a panacea because they need to be validated, right? They need data for training the models. They need data to validate the models. They need data to evaluate the models. And so one way that our group tries to do this is by combining insights from in vitro and in silico approaches to help fill these gaps. And we think of this as a way to prioritize chemicals for further study while also reducing the use of animals. But in the meantime, which is something that a lot of folks that are associated with the Academy and hopefully a lot of the folks that are listening to this webinar are working on, we really, really need good treatment and destruction technologies because we already know that many of these chemicals are out there and people are being exposed. And so we need ways to tackle the problem from multiple perspectives. So before I open up for questions, I just want to acknowledge our collaborators, so Dr. Jennifer Field at OSU, and projects that are funded by CERDIP and Concawe, where we did this protein and membrane interaction work. For the molecular modeling work from Yushin Cao, our SRC industrial liaisons were incredibly helpful in identifying chemicals and procedures. So Brooke Vermos at IBM, Sarah Wallace at Texas Instruments, Dave Speed at Global Foundries, Mansour Wanpour at EMD and Ralph Donnell at EMD, Kai Williams and Tim Eakley at TI, and Bob Leet and Kevin Wolf at Intel. We have funding from Concawe, NSF, CERDIP, and SRC for the projects that I presented today. And I also want to acknowledge the resources from the University of Pittsburgh Center for Research Computing. And with that, I'm happy to take your questions. I was looking at I have often use this final slide and I realized I'm no longer on Twitter, so I better take that out, but you can find me on Blue Sky for the time being. So feel free to reach out if you have any questions we don't cover today. Thanks.
Dr. Ng, fantastic presentation. Thank you. That was awesome. That was fantastic. Exactly what we were looking for as we put this series together. So some questions have come in the chat, and I'll encourage colleagues to continue to put some additional in, but I'm just going to run through kind of some things that we can talk about. And then what we can do is we can kind of answer those, and as additional things come in, we can kind of go from there. So stuff to kind of talk about is like, let's go all the way back to the background, because one of the things about the Academy is that we've got a diverse range of professionals here, right? And so when you're going back and we're talking about a study of toxicology, and you throw out terms like an effect, and then we talk about things like carcinogens and all. So when we're talking toxicology, is this kind of everything under the sun? Is carcinogen kind of a part of toxicology? Or are there other things? Because we are knowing these PFAS are affecting people's, all these different kinds of systems in the body, right? Like what's going on with fertility, maybe what's going on with cancer. So does toxicology cover all that or just some parts? So some people might have some questions in that background. And then, how that differs from epidemiology. And it seemed to me that in a public health model, you were really focused in a lot on the toxicokinetic kind of part of looking at how it partitioned and got to a part in the body. So what's the difference between thinking about the solubility concept of oxynol water versus this idea of docking? So as engineers are getting their mind around it, what's what's the difference and how do we take the other systems we might know, like for example, activated carbon, and how do we think about that versus, docking versus partitioning? So if we can talk about that a little. And then you're using a pretty complex partitioning model, but yet it's still very simplified, right? And so at some point in time, if I add enough variables to anything, I can get it to fit whatever I want it to. So what's that balance between including more and more supposed mechanism, but not really knowing whether it's actually just a fitting exercise or whether the mechanism is really what's playing a role there? And in particular, when we're thinking about scaling from public health down to the granular of individuals, right? How does genetic variation and environmental exposure variation play into the kind of measures that you're creating? Where can we push those into human health, and where are some of the stumbling blocks? We've got a question around PFAS mixtures, right? So some folks were asking specifically, right? When we get into mixtures, do they compete with each other? Do they interact with each other? So are your experimental results all on single compounds, or are they on mixtures? And how did you tease those apart? When you're doing your in vitro approach, right, were these done at these kind of extreme low levels, limits of detections, which was where we're trying to look at regulation and elimination and destruction? Or were these done at more practical levels? And how do you extrapolate the practical data down to the limit of detection for our assays? And then perhaps maybe a few very practical questions, right? Like, I know that I want to drink my glass of red wine with dinner and eat a little chocolate because the antioxidants are good to help me not get cancer. So should I change the fat content in my diet since PFAS seems to be partitioning with some of these? Are there strategies there that might emerge? Another practical question, we don't even know everything that's in petroleum. So we use stuff like, BTEX as a surrogate. So do we really need to study all these different PFAS and PFOS? Can't we just use a couple of surrogates and get away with, as long as these are good, the rest we can let go? And it was awesome, the amount of detailed, kind of clearly toxicology, biological work that was here. Where would engineers pick up some of this, right? So it would make sense for the biologist, but as an engineer, a lot of interesting content here. Do you get that from collaborators? Did you get that from who you had as helping as part of your training, or where do students pick that up? So that's some of the stuff maybe that we can talk through. Does that sound reasonable? There's a lot there to bite off. So
no difficult questions there at all. Yeah,
no,
it's just all
super
piece of
easy.
cake.
Super softballs. So if we can go,
Sure.
yeah, maybe if you want to pull that slide down, then just people can see our smiling faces, if that's helpful.
I can do
All
that.
right, so let's think about this. So back at the high level, right? So, okay. I hear
Should we
terms
start
like
about toxicology first?
yeah, I hear
What
terms
fits, what
like
doesn't?
toxicology. I hear terms like epidemiology. I hear terms like carcinogen. Can you help us tease those apart so that everybody knows the same thing on this webinar?
Sure. So the first thing I will say is that I'm an engineer. I'm not a toxicologist. I just play one on my research. So I will answer this to the best of my ability. And that is, first of all, to say that toxicology is basically just looking at, when an agent has an adverse effect. So it's very, very general. Within toxicology, we have different classes of types of effect. And so carcinogenicity is a very specific type of effect, that's something that can induce cancer in an organism, right? So something that's carcinogenic. And when we talk about risk assessment, we do separate types of risk assessment for carcinogenic versus non-carcinogenic endpoints, just because carcinogenicity is seen as worse because every exposure may increase the risk that someone will get cancer to a carcinogenic substance. Whereas for a non-carcinogenic endpoint, there is really a threshold dose where if you're below that threshold dose, the effect doesn't happen. So that's one of the big subcategories. And then thinking about the difference between toxicology and sort of this partitioning part, the toxicokinetic part, a lot of environmental engineers get into toxicology through studying ecotoxicology, right? And a lot of ecotoxicology starts out being about narcosis because that's the easiest one to explain. And that has a very strong correlation with the internal distribution, because it basically says if something is bioaccumulative, you're going to have enough of it that it's going to disrupt your cell membranes, and then you're going to get this narcotic effect, the non-specific type of toxicity. The more interesting stuff is specific toxicity, and that means that there is an interaction with a specific receptor or something else that causes something above the baseline, above this narcosis, and usually happens at a lower concentration. And so a lot of what I was talking about with specific receptor interactions is about that specific toxicity. So that's kind of the general toxicology piece. And let me put in a plug here because it fits with a later question about why am I doing toxicity as an engineer? So I started, my master's thesis was on doing computational fluid dynamics of a nanoparticle synthesis reactor. I did not start out in this area, but I started looking into ecosystem modeling as a grad student, which started me down the road to getting into biology because I had to understand the life of a fish. What does a fish eat? What does a fish eat when it's little versus when it's big? And so I got into reading a lot more biology, and then getting more and more into bioaccumulation took me down the road of toxicokinetics. And so I never had formal training. And that's sort of what grad school is. It's informal training for everything, right? So I never took a real class on this, but I just read a ton of research papers and then started learning about the tools and applying them myself and then talking to other people that use them. So if you are bio-curious as an engineer, there is a way to get there, right? So you just have to start reading the literature and talking to people, and you'll find yourself down a bunch of rabbit holes, but it's a lot of fun.
Fantastic. No, and that helps just to kind of get us on the same page from the background. So early on, one of the things that was clear that you were looking at was this idea of oxygenol water coefficient, right? So we as engineers can kind of get this. We think about, subsurfaces and NAPLs and where does something partition, so we can kind of get our minds around that. And then we've got, stuff like activated carbon, and we think about, binding sites, and that's a different kind of thing we get our mind around, right? So can you talk about how you're talking about a partition coefficient in this kind of study, and then the idea of docking, and how would we take kind of the broad generic environmental engineering analogies and help people understand the difference between what you were talking about here with partitioning and docking?
Yeah, so partitioning is really absorption into the phase, right? So partitioning is really what you would look at if you were looking at, organic carbon water partition coefficient. So when we do stuff with KDs and soils, that's what we're talking about, right? It's the chemical is actually dissolving into the organic matter of the other phase. And the thing about partition coefficients is that they should be on a straight line, right? It should be, you increase it in one phase, it increases in the other phase, the ratio stays the same, that's your slope, that's your partition coefficient. Docking is trying to understand the specific interactions at a protein binding site, and therefore it is a binding site model. But we use those in engineering all the time too. So we use them when we talk about sorbents, right? So all these sorbent binding site type models work. It looks like Langmuir isotherms, that sort of kinetics. It's enzyme kinetics also, right? So binding to proteins is the same as the way things bind to enzymes. And so you're just seeing something that has a linear range. That's where we get that distribution coefficient, and it's saturable. So you get to a point where you can add more stuff and it's not going to go up. So on a basic level, that's kind of the relationship.
That helps so that folks can get their mind. So now you then are starting to use an increasingly complicated, not because it's not really complex, but an increasingly complicated model of these partition coefficients, right? So let's add in this one, let's add in this one, and then you're doing some fitting. So what's the balance there between adding in mechanisms and simply fitting it to enough parameters that, well, everything fits as long as you give it enough?
Yeah, modelers get this all the time, right? Because they say, well, how am I supposed to trust your model? Because you can make anything fit. And you can, you add a bunch of parameters, but you need to have a rationale behind each of them. And you want to have the least number of parameters you need in order to be able to explain what you see, right? So there are classical means to actually evaluate this, right? So you can use like an information criterion as you make the model more complicated to make sure that you're getting the most parsimonious model possible. I think that in terms of this partition coefficient, we want to have a look at, like, broadly speaking, what are the key phases that are present inside of a tissue. And so you don't want to include every molecule because it's endlessly complex. And you also want to be able to see, does this carry through to different tissues? And if there are tissues that are more similar or more dissimilar, does the explanation in the phases help explain the difference in the phases help explain the differences in the behaviors at a large scale? I do think you need to be cautious of making your model overcomplicated. In our original PBPK model for PFOA in the rat, I think we had 72 parameters, but we didn't fit any of them. They were all just pulled from the literature, and then we saw kind of how it would work. So there's no, so in general, in the models that we do, we try not to do parameter fitting. we try to identify a parameter and look at the value. So I think it's a bit more on, if you're familiar with polyparameter free energy relationships, PPLFERs, that's the more complicated partition coefficients that a lot of engineers are familiar with. And so we're kind of looking at that, but almost at a coarser grain, because that's getting into these molecular level van der Waals interactions and things. But we're just like, okay, well, let's not maybe go down to that depth, but see like which types of, is it a specific binding or is it a partition reaction, interaction, and we use that to figure this out.
Fantastic. And so kind of going down that line of reasoning, right? We do an awful lot of our work and then we'll say, let's rather than look at the whole generic public, let's look at vulnerable populations or susceptible populations, whether it's because of their behaviors and they've got an exposure route, or it's because of their genetics and they've got some predisposition. So as you're factoring this in, where is your work falling in that generic, public versus actually hitting people that might be the susceptible or vulnerable populations? Have we even gotten to that stage yet with some of the toxicokinetics and toxicology around PFAS?
Yeah, we do a little bit of that. So I think the main thrust of the work is just to say, can we understand how these are working in the general case? Because that then helps inform how would we identify the vulnerable population. So one of the things that we did initially was determine that some of these kidney transporters are important for setting elimination rates for certain PFAS. Once we figured that out, we asked the question, well, what happens if you lose kidney function?
Right.
Is that going to lead to more or less PFAS accumulation in the body? And so I had a postdoc who worked on this and kind of looked at different types of kidney injury and how that would impact the body burden for PFAS. And also it helps to understand, because sometimes you'll take measurements in populations and there's a reverse causation issue here, right? You'll find that people who have different kidney function have higher PFAS burdens. Is this because the higher PFAS burden caused them to have worse kidney function, or is it because the different kidney function made the PFAS stick around longer? So it's important to try to piece these things out.
Absolutely. We had a few specific, more, kind of chemistry focused questions, and maybe I'll touch on a few of those right now. We hadn't covered them when we went specifically through it, but they kind of fall in our question line at this space. One of the questions was, what are the industrial domestic sources of side chain aromatics? That's a pretty specific one, but maybe you have an answer to that one for us.
So off the top of my head, I do not remember exactly where they were used, but they're going to be, so they were identified as used in photolithography. That whole study was about photolithography relevant PFAS. So they either came from a patent or they came from some other study that looked at those chemicals. And I don't, off the top of my head, know which particular process they're used in, but it's in our SI. So.
Okay. I appreciate that. It's a great question, but I was thinking, well, unless you have that one on your fingertips, you might not be able to do that one. There's a couple more that are in that space, and we'll just share with you. But when you're thinking about the binding coefficient, how do you evaluate competitive binding, multiple, is your work essentially reporting singles as opposed to mixtures when you're doing this work?
Yes and no. So for the molecular simulations, we do single, because I don't think there is a really good means to do competition in the molecular simulations. What you can do is you can have like the endogenous ligand already bound in the protein and then see where the PFAS would bind. If that binding site is not available, that's one thing you could do. But for the in vitro work, we did when we were doing the membrane water partition coefficients, we tested running the PFAS both as mixtures and individually and found very little difference in the results. This is also because the concentrations were not crazy. So I think this also addresses one of the other questions that we had. So we're looking at a range of concentrations, but they go from rather low, the nanogram per liter range, up to quite a bit higher. But once you get too high, especially for the very hydrophobic PFAS, it just becomes a mess to try to interpret the data because they stick to everything. So you kind of want to stay within reasonable ppb, maybe up to ppm levels, but usually ppb is sort of the safe space for us. But we have done some competitive assays, not for this work, with equilibrium dialysis to understand, this competition with endogenous ligands. So you can do that in vitro, and I think it's interesting. It's something we want to pursue also we're starting to get into surface plasmon resonance as a way to do this in higher throughput, and you can do some interesting competition assays there.
Okay. And then another question that kind of skips from the toxicokinetics to the toxicodynamics, kind of bounce off the two. So are there changes in the amount of toxicity, right? Let me read the question exactly, and then we'll see if we can. How well do binding affinities correlate with toxic effects across different kinds of toxic organics? So you get where
Ah,
I'm going about thinking about the relationship between the kinetic side and the dynamic
mhm.
side. So,
Yeah, I think this also depends on the receptor, and how upstream of the end effect that receptor is, because we see there's been a lot of work looking at PFAS interactions with different PPARs, like PPAR alpha, PPAR gamma, because those are implicated with liver toxicity and some of the metabolic effects.
sure.
And I think those are pretty well established in different types of models. Um, Something binding to serum albumin, not really related to toxicity. Albumin is a carrier, it binds to all sorts of stuff. It's very, what do they call that? Promiscuous in that sense. But it is linked to the bioaccumulation potential. So there's, we look at receptors for different reasons. Some of them are more for distribution and accumulation, and some of them are more directly linked to the toxic effects.
Makes sense. And we did have a specific questions about maternal milk and partitioning there.
Mm.
So not that you were addressing it in your study, but any insights or observations? Is there any literature out there that you're familiar with?
So I've read some. I am not super familiar with it, but what I can say is that there are PFAS can be transferred to maternal milk. My understanding is that other POPs are more effectively so because other POPs accumulate more in lipids than PFAS do. So there's certain PFAS that transfer more through maternal milk than others. But the bigger concern for PFAS is cord blood because they do bind. You find them a lot in the blood, and so they're transferred to the fetus through cord blood.
What I was imagining from the results you were talking about. So one of the things then that this kind of leads into, and this is a little bit of speculation outside of your immediate area, but perhaps maybe you've read some literature in it. But again, this kind of comes to this idea of like diets and behaviors and things we do, right? So, if I drink my glass of red wine, I'm preventing myself from getting cancer. I eat a little dark chocolate, or, and it makes you feel good that you're doing something healthy, whether you really are or aren't, but you at least think about these things like antioxidants and
Yeah.
diet. And so how about PFAS in diet or PFAS in activities? I mean, yes, we've seen the please get rid of all your nonstick, cookware, or we see those kind of things that are
Right.
out there, but are there dietary things that we might be thinking about or places where people are starting to work on that?
Yeah, this is where it can be really tricky with PFAS. With POPs, you could say like go for lean meats, take the skin off your fish. It's not as simple for PFAS. And one of the frustrations is we've done some work on like PFAS in seafood, and it doesn't always seem to be rhyme or reason in which species you'll find a high, we find really high PFAS concentration in some clams, and we couldn't really, or smelt.
Mhm.
So it's harder to make that kind of recommendation for PFAS. Except I will say, maybe skip the liver and onions because liver is definitely a big, not just for PFAS, but for a lot of chemicals.
A
It's
lot.
meant to be detoxifying your body, so it's maybe not great to eat it. And in general, where we don't have a clear direction, I generally tell people to lean into your omnivory in the sense that try not to eat the same thing all the time, because if it just so happens that you pick one of these problematic things, you're going to be getting a really big dose. And so by trying to vary the trophic level and the source and the type of foods that you eat, you're kind of insuring yourself against having accidentally picked the worst thing.
Yeah, makes sense. Makes sense. Yeah, I've heard people refer to the liver and onions as kind of like having your picnic lunch at the landfill, right? And you're like, okay. Now, we still don't know everything in petroleum, right? So BTEX and we use surrogates and things like that. How about for PFAS? Where are we at? I mean, it feels like we're kind of like it's the boogeyman, and let's measure all of them and let's go after all of them. Is it really that bad, or are there maybe some that are starting to emerge as, hey, these are the canary in the coal mine. This is the
Right.
equivalent of BTEX, and let's go after these and not worry about everything else.
Yeah, I think there's a ton of interest in this right now, and people are trying to figure out like what would be the representative PFAS. I don't know if we have that answer yet, but I think that the question depends on how you're going to use that information, right? So, for example, let's say you want to use it to prioritize cleanup. Then maybe what you want to do is not target the PFAS that is like the most representative of an effect, but really the PFAS that if you get rid of this one, you'll have cleaned up most of them, right? So I think that's even an easier question to answer. For understanding the effect, I think some of our results point to there are some key differences across structural groups. So you may not need to understand everything about every PFAS, but maybe pick a representative for key structural groups. Like you're going to need some sulfonamides, you're going to need some ethers, you're going to need some carboxylic acids, and that's how you can try to build it out. And then within the groups, as you go across the chain lengths, you might be able to make predictions, right? But sometimes going across the groups is a little bit difficult.
Okay. Okay. And so there's a question that's kind of along this line that I'll just kind of read verbatim, and I think we can infer your answer from it. But are there any studies comparing blood levels of PFAS and related chemicals to specific kinds of cancers or other health effects, right? Are there any particular ones that are the, hey, this is the really bad one, or this is the, and I think that's kind of what you're alluding to in your answer there. I don't know if you want to elaborate any further.
Yeah, I do think that so in terms of cancer, the most data that we have are for PFOA and PFOS. And I don't know if they're out at the moment. If they're not, they're soon to be. The IARC, which is the body that basically nominates substances to be considered as carcinogenic, have their monographs coming out on PFOA and PFAS. If you're interested in the scientific evidence for their carcinogenicity, that's what I would look at. Okay. And also everything that came out of the C8 science study, specifically for PFOA, has a lot of that evidence in it from epidemiological studies.
Fantastic. And then, like you said, trained as an engineer, now working in this space that some people might accuse you of being a biologist, right? So what's your advice for folks that would be either perhaps even undergrad students listening to this and thinking about trajectory of their career, or folks that are at a stage where they're saying, I'm looking for something else to get into, or I'm looking for a new challenge, or I'm looking for an additional challenge. What would be your words of wisdom for people who are saying, hey, I'm interested in this, maybe PFAS and toxicokinetics directly, or maybe just becoming more knowledgeable in this area and understanding this technical aspect a little bit better? What's some advice for them?
Well, advice number one is read. And it's something that as a faculty member, I really miss because I haven't had the chance to do a lot of that other than papers that I'm reviewing. So that's something I'm really looking forward to in my sabbatical, is to be able to sit down and actually read more stuff again. There's a lot of back and forth and conversations in the literature, and especially if you go back to some of the older stuff, it's really enriching to understand how people figured things out and then try to apply that to your system. The other is just don't be shy. Go out and if you're an undergraduate student, go talk to the faculty in your school, in your department, in the next school across the street. Say, hey, I want to do a semester of research. I want to do a summer of research. I want to find out more about this to see if you really like it. A warning I give to all new PhD students in my group is it's really hard to get a PhD. You get so little external, praise for the stuff that you do. So you have to be really motivated by the topic. You have to find the thing that fascinates you, because otherwise it's going to be miserable. So it's important to just try to get these experiences and read a lot and find out what excites you. Yeah.
Well, Dr. Ng, that was a fantastic presentation today. We're so grateful for you taking time out of a busy schedule. Congrats on the sabbatical coming up. Enjoy that. Thanks. And we will turn it over to Marissa to close us out.
Okay, thanks everyone for all your questions, and thank you to Carla for sharing your knowledge with us today. We have several webinars planned in the near future. You can go to aaes.org/events and reserve your spot. Don't forget to register for the 2 upcoming PFAS webinars scheduled for January 22nd and January 29th. If you're not yet an Academy member and you're considering joining, you can email me to discuss our membership options. For all board certified individuals, PDH certificates for this event will be sent out by the end of the week. That's all for now. We enjoyed being with you all today. Thank you so much and have a great day.
Bye.